This is the fourth of our special topics lectures, and I'm very pleased to welcome Dr. Joel Wertheim. So Dr. Wertheim and I have been working together for a couple years now. Dr. Wertheim works a lot in HIV molecular epidemiology. He's also done a lot of work in the evolution of other viruses. Please, take the floor, Joel. Thank you so much for joining us.
All right. Thank you all for having me. Thank you, Nima. Dr. Moshiri. It's a pleasure to be here. As you mentioned, I'm a professor, Joel Wertheim, and I am at the UC San Diego School of Medicine. I've been working here for about 15 years now. I've been a professor here for about 11. And my focus, well, first of all, my training. I'm an evolutionary biologist by training, even though I happen to be in a medical school. I look at how organisms change and adapt over time. And the organism that I am particularly fascinated by are viruses. In this case, we have SARS-CoV-2 viruses. And today I'm going to talk to you a bit about viruses, their evolutionary biology, and how we can use computational approaches to understand their evolution history. Specifically, we're going to focus on the origin of viral pandemics, and two of them that I've spent my life studying, HIV and SARS-CoV-2. But first, I'm going to give you a little bit of technical background. Nima assures me that sort of the cream of the crop of students. So even though you might not be familiar with this concept, it is one that is near and dear to my heart and to my research, and it is called the molecular clock. And it was invented, or essentially discovered, by this gentleman here, who, if you've taken any introductory chemistry classes, you may be familiar with. His name is Linus Pauling. He is most famous, actually, for being the only individual to win 2 Nobel Prizes, where one of them was not in the field of scientific research. So his first Nobel Prize was in chemical bonds, and the second one was in nuclear nonproliferation, trying to denuclearize the world. But despite the fact that that gentleman won 2 Nobel Prizes, he was not always right in his scientific hypotheses. For example, he was a big proponent in what was known as the triple helix for DNA, which, if you've taken any biology course or seen any of the Jurassic Park movies, you may be familiar that DNA is a double helix, not the triple helix, as Dr. Pauling suggested. So even the greatest scientific minds are occasionally wrong. And in Linus Pauling's case, sometimes doubly wrong, because if you've ever had a cold or the flu, and someone says, are you getting enough vitamin C? Well, that was also one of Linus Pauling's ideas, especially later in life. And in fact, he's the reason that we have airborne, the cure for the common cold, and all of these other suggestions that vitamin C could cure all of our maladies. They can't, and he stuck with that one to his dying day. At least he let the triple helix go in the 1950s. But vitamin C, we're still living with today. Anyway, but importantly, he also had great contributions to the field of biology in the form of the molecular clock. Now, what is the molecular clock? It is basically just the steady accumulation of mutations over time. And we're going to talk an awful lot about mutations today, so you should get familiar with it.
Some kind of mutation.
Like a mutation.
Cordyceps mutated. Was some sort of mutation.
Mutated them into horrific monsters!
A mutation.
Why? Because we are mutants.
The pangolin has mutated how?
Any mutations, augmentations?
Mutated the dinosaur genetic code.
They are mutated, Seabass.
Are mutations caused by radiation?
All through mutation.
That better not be the mutants.
Okay, so Hollywood gets a lot wrong about mutations. So I'm going to explain to you exactly what they are and what they're not. So if you think of a stretch of DNA or RNA, as the case may be for a virus is, they're comprised of nucleotides, A's, T's, G's, and C's. All a mutation is the change from one nucleotide to another. So in this case, let's say we have sampled a viral genome in year 1. Let's zoom in a bit on this one. And you can see here in year 1, we have a sequence, and then after about a year, maybe the virus will mutate, and you can see here we have a change from a G to an A. And then in the next year, the virus might mutate again. Now we have a change from a C to a T right there in the middle of our shortened, truncated viral genome. And this is the molecular clock in action. These are just neutral changes over time. They're not necessarily impacting how good the virus is at infecting you or avoiding your immune system or making you sicker or less sick. These are just sort of random changes to the virus's genome that we can use to track its evolution over time. By year 4, we get another mutation. In year 5, now we've gone from a T to a C at the very beginning of the genome. And then by year 6, we see we accumulated our 6th and final mutation, or 5th and final mutation in the middle there, going from an A to a G. Now, the trick is, for most viruses, we don't actually have every single intermediate form sampled. So we're kind of stuck with our first virus, and then again, in year 6, we have another virus, and we sort of infer the number of changes that have occurred over time, essentially trying to count the mutation.
Some kind of mutation.
Not do that whole thing again. Okay. So as a great example of this, one of the most clock-like viruses we have is influenza or influenza A viruses, which causes the flu. And we've known about the molecular clock in flu and how consistent it is for a very long time now. This paper is from 1991, which even predates my entry into this field. So we've known about this for a while. What you can see here is, as you plot, flu's been circulating in humans since, well, this version since the Spanish flu in 1918. And we've been sequencing it over time, pretty much every year, going back to the 1930s. In this case, through the 1980s. So it's 50 years of sampling. And you can see that there's a relationship between the year of isolation, when that flu virus was circulating in humans, and the number of nucleotide substitutions or mutations since the root. And we would call this a correlation, where there's a linear relationship between how many nucleotide changes have occurred and what year that virus was sampled in. You could even get a pretty good idea of what year the virus is from just from looking at its mutation. And to show you how this progresses through time, we can go back. So in the 1930s and 40s, you would only have between 10 and 20 nucleotide substitutions from the sort of ancestor of that tree. By the 1940s, now we accumulate another 10 substitutions. And as we move forward into the 1950s, another 10 substitutions. And you can see that as we move forward in time, we accumulate more and more and more nucleotide substitutions as we sort of move up that y-axis. And then by the 1970s and then into the 80s. In fact, if we were to extend this up until 2009, when this particular strain of influenza disappeared, when a new pandemic strain, the 2009 H1 swine flu sort of, well, actually, no, this particular one is still around today. They'll get vaccinated. So this evolution would have continued up until 2024, and will go into the future, accumulating more and more nucleotide substitutions and more colors of the rainbow. That was clear. If not, anything's unclear, please put it in the chat. I'd be happy to. Okay, so that's sort of the basics of the molecular clock, just counting these mutations over time and trying to figure out when did certain viruses ancestor. Now for HIV, we can use this to answer a very, very important. That is, how did the HIV pandemic? And I'm going to put forth 3 of the most popular, if not scientifically robust, hypotheses as to the origin of it. And they are, but in order to understand those, we first need to understand what phylogenetic trees are. Now, Dr. Moshiri has informed me that you got a little bit of background on phylogenetic trees, so this should all be nice and easy review for everyone. We're going to do it soup to nuts. Okay, this is phylogenetic tree. This is something that we extract from DNA or RNA sequence, and this tells us the relationship of all life on Earth. And you can see here the phylogenetic tree is comprised of nodes, which are the organism at the tip. And here we see a bunch of great apes, including humans. Now, highlighted there is Charles Darwin. But Charles Darwin is a young man before he had the big giant beard, because it's important to remember that not all scientists are born with big bushy beards. Here he was very clean shaven. This is when he was on the beach, a young man. But we also have other species here. We have chimpanzees, we have bonobos, and we also have gorillas. And those are all nodes on the tree. And as we move back into the tree, we see branches. So we see the branch that leads to humans, the branches that lead to bonobos and chimpanzees, the branches that contain those internal branches that represent the ancestors of bonobos, chimps, humans, and gorillas. Okay. So, I have a question for you. Which species on Earth is our closest living relative? And either you can unmute yourself or you can put it in the chat and I'll read it out right now. But just give me that answer. You've had a handful of years of school. You must have at least some guess as to our closest relative.
Any guesses from
Anyone?
folks? Feel free to type in the chat. Feel free to unmute.
Chimpanzees. Fatima. Excellent guess. Anyone else? Tell you the answer. Okay, so we're going to go with chimps. I like that guess. Okay. So in order to figure out who our closest relative is, we have to figure out who do we share a common ancestor recently. Well, let's start in our tree here. We have Charles Darwin representing all of humanity, at least a painting of him. Go back in time over hundreds of thousands and millions of years until in that phylogenetic tree we hit an ancestor that we share with other living. And when we hit that ancestor, then we turn around. Oh, sorry, we hit that ancestor, which I've marked here with a star. That's where that's going to tell us the answer. And then we start moving forward in time, and we see which animals get to. And as suggested, we get to chimpanzees. So chimpanzees are our closest living relative. But so are bonobos. And in fact, bonobos are another ape that lives in sub-Saharan Africa. They live south of the Congo River in the Democratic Republic of the Congo. They're the ape that tends to resolve their disputes through sexual activities rather than violence. I'd like to learn more about them? You're just Googling. We're not going to spend that much time on them today. It's mostly viruses, but this is a good example. You can see here that humans are just as closely related to chimps as they are to bonobos because we share a common ancestor at the same point in the tree. And in fact, bonobos and chimpanzees are more closely related to each other than either of them are to humans. So we don't have one closest relative in the animal kingdom. We actually have two, bonobos and chimps. All right. So where did humans come from? So we can learn more from the tree than just who we're related to, but we can learn where did we originate. Let's do that here. So we're going to take our same tree as before, but this time we're going to start labeling it with countries. Now, we know that chimpanzees are found all over sub-Saharan Africa, really in that central band, all the way from West Africa. Bonobos are found just in a very small part of the Democratic Republic of the Congo, and gorillas are also found across that band across Central Africa, mainly in West Africa, all the way over to Uganda and Rwanda. Now, Charles Darwin, on the other hand, he hailed from England. So we're just going to give him all of Europe here, just as a representative European. And here we can say, where did humans come from? So we can start recreating the geographic history of all of our ape ancestors on this tree. So if we start with chimpanzees, we go back in time, and we start with bonobos and we go back in time, we can see that they share a common ancestor very recently, about 1 million years ago. About 2 million years ago. And since they're both found in Africa, we can infer that the ancestor of chimps and bonobos was also found in Africa. Pretty good assumption. They both are still around DRC. But what about gorillas? Well, they're also found in Africa. So if we go all the way back to the root or the base of our tree, we can see that, oh, that connects with chimps and bonobos as well. So that was probably in it. And that means that that internal node there that represents the ancestor of chimps, humans, bonobos, and gorillas was also probably in Africa. And then as we start moving forward, we see that humans must have at some point been in Africa and then left to basically colonize the world. So we can see that humans most likely originated in sub-Saharan Africa and then traveled to Europe, Asia, Australia, and the Americas. Especially every continent except Antarctica. Which means all humans can trace their ancestry back to Africa, which the fossil record supports. And we can infer that based on the phylogenetic tree alone, even in the absence of any fossil found anywhere else. But how do we know? Well, that tree that I just showed you, the original one that showed who humans were most closely related to, was constructed using mitochondrial DNA, that small little genome that's passed along from mother to offspring to mother to offspring to mother to offspring. And you can also do the same thing with the human genome with its 3 billion nucleotides, which according to classic movies, you could watch this screen here for days and still not see all those A's, T's, G's, C's. Fortunately for you, we're working on viruses. And viruses have a much, 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 much smaller genome. In fact, this, for HIV, is its entire genome. Let's just take a look here. Memorize this. We'll have a quiz on it later. It's about almost 10,000 nucleotides. But that's it. You can see every A, T, G, and C, or in fact, A, U, G, and C, because they have uracil, in the HIV genome. And it can all be visualized on a single slide. It's pretty small. And in fact, the part of the HIV genome that we're the most interested in today is probably just this 1,500 nucleotide stretch right here in the polymerase region, which allows it to replicate itself and insert itself into the host. And in fact, this is what HIV genome looks like up close. This is what we call an alignment of a whole bunch of different strains of HIV, and you can see all of the mutations that separate. This is an actual real-life alignment that I've used in research. These are HIV sequences from San Diego County, from people living in the county. And we can use sequences or sequences like these to ask the question, how did the HIV pandemic? Because now you've learned all about the molecular clock, you've learned how to read a tree, you've learned how to figure out who humans' closest relatives are and where they come from. You should be able to do the same thing with a virus. And I'll show you right now that I think you. All right, so let's get back to our 3 hypotheses on where HIV comes from. First, is that during the development of the oral polio vaccine in the Eastern Congo, somebody grew up a polio virus on chimpanzee kidney cells. Those kidney cells happen to have another virus called the simian immunodeficiency virus found in chimpanzees, and that virus was able to jump into humans as a part of this vaccine trial vaccine development in the late 1950s, early 1960s. Another very popular and perniciously persistent hypothesis for the origin of HIV that the CIA created in a lab, likely in Fort Detrick, and then released it upon the American populace as a way of sort of controlling minorities. And this was a very popular hypothesis. It even made it onto the evening news in the late 1980s, early 1990s. I understand the idea of the evening news is somewhat anachronistic, but back then it was a really big. And also, there's this other theory that it was a zoonotic jump from chimpanzees, potentially while hunting and butchering them for food. And this could have happened at any point in time. But let's put some dates on our first two hypotheses. Oral polio vaccine means HIV should have originated in the 1950s. CIA did it. Well, the CIA didn't exist until after World War II, so we're going to put an upper bound there in 1947. Okay, now this idea that it was the oral polio vaccine actually got its start in a less than reputable publication, Rolling Stone, who in 1992 published this article here on the origin of AIDS, suggesting that it was from this polio vaccine trial. The claim that the CIA did it is a little more pernicious and a little more interesting for our modern times, because it really a great example of how the disinformation war that's currently being waged across our country and across the world is nothing new. And I strongly encourage you to Google the words Operation Infection sometime later today. Infection with a K. And just read up on this. There's a great YouTube video that goes through all of this. But essentially, the idea that the CIA created HIV was not something that just paranoid tin hat conspiracy theorists came. It all traces back to a single news article that appeared in an English language newspaper in India in the 1980s that said the CIA developed it and it was done in an American army base, and we have evidence. Now, this English language newspaper in India was known to be owned and operated by the Russian KGB. And they let it sit there for a few years, untouched, until in the late 80s, early 90s, they started releasing articles in Russian saying, oh, we think the CIA did it. And they pointed back to this original article. Eventually, everybody had a citation trail that went back through these various news articles and then scientific reports. But it was all based on one, frankly, fake news article that was planted in a KGB operated English language newspaper in India. But nobody bothered to go all the way through and document, which is how it ended up making it onto the evening news and into newspapers in the United States, without anyone really being able to fact check its ultimate source, which was Soviet propaganda. All right, but how can we disprove it with science? Because just knowing the truth sometimes isn't enough. You need science. So you can take the molecular clock and you can build that same sort of correlation that we showed earlier with influenza, where you look at the date of sampling for a virus and how many mutations that virus has from the root of the tree or its branch length. And when they did this, what they found is that the ancestor of HIV likely originated in the early 20th century. This paper suggested around 1928. Other papers more recently have suggested 1919. But the point is, it had been circulating in in sub-Saharan Africa for quite some time before it was discovered. And in fact, they found a single virus sampled in 1959 in what was then Zaire, now the Democratic Republic of the Congo. And if you mapped the depth of that virus in the tree onto your molecular clock correlation, you could see that it looked like it should have been sampled around 1957, which is just about where you'd expect to find it, which means that our molecular clock in HIV does tick reliably and can let us know that HIV was circulating, likely circulating in sub-Saharan Africa all the way back to the early 1900s. So, if it's circulating in sub-Saharan Africa in what was then Zaire in 1950s, but also likely we can infer 1920s, well, we can get rid of the oral polio vaccine hypothesis. And we can also cross off the CIA did it hypothesis. And all we're left with is this zoonotic jump from chimpanzees to humans. And in fact, when other scientists went in and sampled virus from chimpanzees living in southwestern Cameroon, shown there in red, Cameroon borders the Democratic Republic of Congo, you can see that those chimpanzee viruses are very, very closely related to the human immunodeficiency virus that's HIV-1 group A that went pandemic. Sorry, group M that went pandemic. So that small population of chimpanzees living in the corner of Cameroon likely were the source population for pandemic strain of it. Thanks to molecular clock and phylogenetics, we have a pretty good idea of how this pandemic started. Now, we don't get to observe the cut hunter that happened over 100 years ago, but we still have but all of the evidence seems to line up to say that HIV was a natural zoonotic origin. I'm going to take a little time here and show you how HIV got into the United States and when did it get here, using all of the same tools that we learned. And here we have an HIV phylogenetic tree where the samples are colored by where in the world they were sampled. We have samples from New York City, from Georgia, from California, a whole bunch from the Caribbean, specifically in Haiti, and then at the bottom, sequence Africa. Now, if we just look at the top part of this tree, we can see that represents how HIV was in the United States in around the 1970s. You can see that the same way that we constructed the ancestor of all great apes, or of the African apes to be in Africa, we can reconstruct that the ancestor of HIV in the 1980s was in New York City, because we'd sort of color those ancestral nodes blue based on how common they are. And you can see that all of those viruses in the US share a single common ancestor, meaning that it got into the US and then exploded around, say, 1970. The question is, where did it come from? And when we can look here, you can see that its closest relative here are in the Caribbean. In fact, within the Caribbean, in Haiti in particular, you have a lot more diversity, a lot that goes back even deeper in time into the 1960s, suggesting that the virus went from Africa in the 1950s and 60s into the Caribbean, where then it diversified, and then it made a jump into New York City sometime around 1970, where it then circulated undetected for 10 years, before anyone had any idea that HIV or even AIDS was. From that, we can hypothesize that the virus jumped into Haiti, and then from Haiti, it made its way into New York City, and then from New York City, it spread around the rest of it. And sort of that same approach that we took to identifying that humans originated in Africa, we can see that HIV originated in Africa, sort of took a skip and a jump through Haiti, and then landed in the United States. All right. That's HIV. Any questions before moving on to the fun stuff? All right. Now we're on to SARS-CoV-2, a virus that I've been working on pretty intensely for the last 4 years, and got me working even closer with your teacher, Dr. Mokiri. So that's the big question of the day. How did the COVID-19 pandemic? I could talk about this for hours, but I'm going to give you all of the abbreviated. So the first cases were known to be associated with the Huanan wholesale seafood market in Wuhan, China, a market that it was believed to be selling wild game for food consumption and also keeping live animals on the premises, which is something that there were only about 3 other markets in Wuhan, a city of 11 million people, known to be doing at the time. But when the WHO, the World Health Organization, went to investigate this in January and February of 2021, they concluded, highlighted it here for you, that there were no verified reports of live mammals being sold around 2019. Now, this is important because the SARS pandemic, which happened in 2002, 2003, 2004, was started by infected animals, specifically palm civets, which are a kind of cat, that were being sold for food consumption in southern China. As SARS-CoV-2 is somewhat closely related to SARS-1, it's thought, well, this may have been a similar route. But the WHO went in and they said no mammals were sold there in 2020. Everyone sort of shrugged, or a lot of people, including myself, sort of shrugged their shoulders and were like, oh, I guess it wasn't live animals. And about 3 months after that WHO report came out, some scientists from China, the US, and Europe published this paper entitled Animal Sales from Wuhan Wet Market Immediately Prior to the COVID-19 Pandemic, where they were tracking tick-borne viruses and just looking to see how many ticks and how many mammals were being sold in Wuhan. And this paper, they took pictures of a whole bunch of animals that were being sold at them. They have snakes here, they have hedgehogs, and in part D, they have one particular animal, zoom in here, known as a raccoon dog. Now, a raccoon dog is not a raccoon. It's actually closely related to a fox, and it's sold for fur and for meat consumption. And it actually is an animal that was known to be previously infected with SARS-1 during the SARS pandemic in the early aughts. So the WHO said it wasn't there, but here we have photographic evidence of these animals being sold immediately prior to the COVID-19. What else is? Okay, so a bunch of us scientists, I count myself in this headline that says a group of scientists took this finding plus a bunch of others to say, you know what? I don't think SARS-CoV-2 came out of a lab. We think it was a natural zoonotic origin, likely at the Huanan Sea. And we pushed back because, of course, every virus comes with its own lab leak story. I just showed you and described to you in detail how the HIV lab leak story came about. But in fact, there's nary a pandemic or epidemic that doesn't come with its own tinfoil hat. All right, so that led us, you know what, I think we can do this and put this whole thing to bed, which led to a paper that we published entitled The Molecular Epidemiology of Multiple Zoonotic Origins of SARS-CoV-2. Kind of gives away the punchline in the title there. But this is a paper that with a whole bunch of scientists from around the world, but highlighted here are Dr. Nima Moshiri and myself there at the end. So this is some work that. Okay, And I'm going to tell you everything you need to know to understand why we think that this virus originated in a market in November 2019. So we took all of the SARS-CoV-2 viral genomes sampled in late 2019 and early 2020, and we performed a molecular clock analysis. What you need to know about the early days of SARS-CoV-2 is that there were two main lineages. This is before all the variants, before the alphabet soup, before the sort of now the obscure variant names that we see. In the beginning, there was just lineage B, which the earliest documented case was on the 10th of December 2019, where someone became sick and eventually was diagnosed with SARS-CoV-2. And every single case at the Huanan seafood market in humans was lineage B, which would suggest that maybe lineage B was the progenitor. But there's also lineage A. Now, lineage A wasn't seen directly in any of the people infected at the market. And in fact, it wasn't even seen in any humans until later in December 2020. But these were people who were near the market, someone who was staying at a hotel nearby, and someone who resided just a kilometer or two from the market, but they didn't work there, and they couldn't trace it back directly to the market, which makes you think, okay, maybe B started circulating in the market and then A popped up. But there's one really weird thing. This is sort of all you need to know about the early genomics and genetics of SARS-CoV, is that lineage A is 2 mutations closer to the bat virus ancestor and relative of SARS-CoV-2 than lineage B. So if you just had to just plot it out and go from bat, mutation, 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 you would hit lineage A 2 mutations before you hit B, which suggests that A should be the, because it looks the most like its bat relative. Here we have to turn to our good friend and SARS-CoV-2 has a very, very good clock-like signal. You can see here that correlation between the number of mutations and time, and you can see that it just clicks, the clock ticks fairly regularly. And when you examine the molecular clock at the beginning of the pandemic, you see something really weird. That lineage B seems to be 2 mutations ahead of lineage A if it were to be the root of, which is strange. Because lineage A kind of looks like it should be the root. It's the most like the bat virus. But lineage B, shown here in this lovely teal color, has about a 2 mutation head start. So why is that? And does that really supported by the whole molecular clock analysis? Yeah, so it looks like B had a head start. Thanks. But is lineage A still the ancestor? So we can go and do that same type of inference as before. Instead of asking what country is at the base of the tree, we can ask what mutations are at the base of the SARS-CoV-2 phylogeny. We apply the molecular clock to all of the early SARS-CoV-2 genome. And here I'm plotting the relative probabilities that B is the ancestral. And what you can see there is that about 80% of the time, or molecular clock analysis suggests that B is the root, and only about 1.5% of the time does it say that A is the root, which for those of you who are really into your statistics, Bayesian statistics in particular, it's a Bayes factor of about 48, which is strongly suggestive that in fact B should be the root of all of the pandemic. But so the molecular clock doesn't like lineage A or the bat-like lineage at the root of the tree. But what if we help it along and we say, you know what? We have the molecular clock, but we also know what the closest bat sequence. Bat viruses, they're recombinant, which means that they're swapping bits and pieces of their genomes together all the time. So there isn't one evolutionary phylogenetic tree to describe them all. But if we look across the genome and we say, all right, what bat virus does it look like here? What bat virus does it look like as we move across the 29,000 nucleotides of the SARS-CoV-2 genome? We can say we think this is what the closest bat virus should look like. And if we put that at the very base of the tree, then we let the molecular clock go again, we find that everything flips. Whereas before B was the favored route, now lineage A. And it's very strongly lineage A, 77% to 8%, which means the bats are telling us lineage A is the route, but the humans are telling us lineage B. They'll suggest that B got a head start, but how could that happen? Why is the ancestor not supported by a molecular? Well, one hypothesis is that it was actually two introductions. Is that lineage B jumped in first from an animal at the market, and then maybe a week or two later, lineage A got introduced. Now, I admit that the suggestion that a once in a lifetime pandemics are about a once in a generation thing of this magnitude, but a pandemic of this magnitude, the world hadn't seen since 1918. So really, once in a lifetime, once in a century event, a highly contagious, deadly pathogen gets into humans. And I'm suggesting to you right here and right now, and in the scientific literature, that that once in a century event happened twice, the same place, in a matter of weeks. But of course, that's very unlikely, because you need like 9 misfortunes to get one zoonotic pandemic. First of all, you need a virus that can replicate in bats. Then it has to evolve ACE2 binding, which is the human cell receptor. Then it needs to evolve what we call furin cleavage, which would allow it to replicate in human lung cells. It needs to get into a secondary host, and it needs to actually be able to transmit that secondary host and not have it be a dead end. Then that secondary host has to get into very close proximity to humans. It has to be able to transmit from that secondary host into humans. Then it has to be able to replicate in humans, something that most of these viruses certainly wouldn't be able to do. And it has to be able to transmit between humans efficiently as a highly contagious pathogen.
But that will never happen. 3 misfortunes, that's possible. 7 misfortunes, there's an outside chance. But 9 misfortunes, I'd like to see that.
Well, that's discouraging. Times like these, you have to turn to the patron saint, zoonosis, George Clooney. I know you don't think that's his job, but in fact it is. George, what do you got for it?
Right. Who are you looking for? Tenzing Norgay. Tenzing Norgay.
Tenzing Norgay. Did he work at the Huanan Seafood Market? He patient 0?
I doubt it. Tenzing Norgay was the Sherpa that helped Edmund Hillary climb Mount Everest.
Let you all in on a secret. The whole concept is conditional probability. Something that even a lot of scientists don't quite understand. Because you see, humans have lived in the foothills of Mount Everest, the world's tallest mountain, tens of thousands of years. All that time, a single human being ever reached that until April 29th, 1950, when 2 humans reached the summit on the same day. Because you see, for Edmund Hillary to get to the top, he needed the right gear, he needed the path, he needed the right weather, he needed the determination. Once you have all those, getting another person to the summit, that just follows naturally. Edmund Hillary and Tenzing Norgay getting to the top of Mount Everest were not independent events. In fact, they were conditional upon each other, and neither would have been able to do it without the other. And once you know that one of them got up, it seems almost a given that there was another. Same is true of zoonotic jumps. Because once you have SARS-CoV-2 circulating in humans, well, then you already know you have a bat virus with binding and furin cleavage that could replicate in human lungs. It already infected the secondary host and was already being able to transmit among those hosts. And in fact, you already know you've had exposure to humans. And you know that after that exposure, it can jump, you know that it can replicate in humans, and you know that this virus is highly transmissible. So in order to get that second zoonotic jump, the only thing that you need is continued exposure to humans. And that's what we had in the Huanan seafood market between November and December 2018, before it was closed and all the animals were. So it's not separate probabilities of another pandemic virus. It's conditioned only on the continued exposure to humans in a wet market. So, but that hypothesis makes a prediction. It predicts that lineage A shouldn't have just been around the market. It actually should have been found in the market itself. Well, sometimes science works properly. We made that prediction. Then shortly thereafter, scientists in the Chinese CDC put out this paper. They pre-printed the paper and eventually it was published. What they found was that on the western side of the market, right next to where all of the raccoon dogs and other animals were being sold for meat, they found a pair of discarded human gloves that had lineage A virus, showing that lineage A was also in the market the whole time, just potentially at lower levels, and that more people had B in the market than A. Leads us to this hypothesis about how the SARS-CoV-2 pandemic. You had a bat reservoir, it jumped into an intermediate host at the market, and then made two separate jumps. Lineage B jumped in first, and then lineage A jumped in, both at the market, and lineage B started evolving first in humans, and therefore accumulated those extra. That's how we think COVID-19. And, got some, Nima wanted me to highlight our press coverage here. So here's a article in the New York Times that said new research points to the Wuhan market origin as origin. This actually was a front page story just above the folds. We were all very, very proud of it. Nima's making me do a victory lap. And with that, I want to say thank you. And on behalf of the Molecular Clock, it's been a pleasure.
Awesome. Thank you so much. Any questions from folks? This is a one-time opportunity to talk to one of the world experts on this subject. So I know everyone's been facing COVID. Everyone's known about HIV. Dr. Wertheim's an expert. Questions from folks? Feel free to type in the chat. Feel free to unmute. No? I guess maybe I have a question then. So what's next? How do we fight future pandemics using the techniques you taught us?
Well, so one of the big differences between HIV and SARS-CoV-2 is that when HIV first started circulating in humans, sequencing technology was barely. Back in the 70s, they had to use radioactive molecules to do sequencing, and now we just do it with light, basically flash drive attached to a laptop. So the sheer number of viral genetic sequences that we are accumulating now is astounding. I mean, we had, I think it's 17, 18 million SARS-CoV-2 genomes. I also study Mpox, monkeypox, and we have tens of thousands of those. HIV, we have millions of sequences wide. it really is an astounding amount of data that people with just a little bit of computational skill, and I mean not as much as Nima have, like people who have as much as I do, which is much small, uh, can really do sort of big work with that. Now, I remember I went to a biology lecture when I was in high school. I'm not going to tell you the year, but it was right before the human genome. And the guy got up in front of us and said, right now, we have more genetic data than the world's scientists can. And you could make a career just analyzing publicly available genome. And it was true then, and it's true now by orders of magnitude. So there's so much we can learn from biology just based on what we see. And I mean, my chosen field, of course, viruses, because I think they're the most interesting and they evolve the fastest. So they're the most exciting to study in real time and track. But that's not to say that, everyone needs to study